turn to the slide that says karyotypes, and there's two examples of karyotypes. We'll finish up by reviewing that, then we'll move on to some actual genetics. So, question? Mateo? I don't know. Dan?
There was this lady that thought that if you were to have twins, it would take you 18 months instead of 9.
Yeah.
Who said this? I don't know.
All right, so let's look at this karyotype here. First of all, karyotypes in general, what are they showing? What is it a picture of? Chromosomes. It's a picture of the chromosomes that a person has. They're arranged in order from largest to smallest, the first pair to the 22nd pair. Is this person on the top, is that a male or a female? Raise your hand if you think you know. Sammy? How do you know? Yeah, now they're not labeled in this one, but you are correct. So how do you know which is which? David?
They're the same size.
Yeah, they're the same size. So the two of the same sex chromosome. This is a female. About this individual.
Male.
Male or female? Male. Any abnormalities?
Down syndrome.
Down syndrome, and how do we know that?
3 21st chromosome.
3 21st chromosomes.
Said that.
I have a question.
Yes.
If they found it out real early, could they like take out?
They would have to find out in the very earliest, like embryo stages, which they cannot really do at this point. So no.
But they will soon.
I don't know.
Maybe.
That would mean you have to, because you can't, you can take, we talked yesterday about in vitro fertilization, you can check with an embryo growing in a petri dish and check, but you can't do it when it's actually in the womb, implanted in the uterus, and a growing embryo. There's an interesting book, I think it was written in the 1950s. It's called Brave New World by Aldous Huxley, and in this futuristic novel, most individuals are born in artificial wombs. Women have chosen that they don't want to carry their children naturally, so they've developed these artificial wombs, and people are like genetically engineered, and they grow inside of this womb, but then some people choose to like go the unorthodox way and have natural children. So it's interesting. If you're looking for something a little different to read.
Very, so
Just don't do it.
if there's, it doesn't say masculine symmetry, it's because they're even.
Because they're the same, yes. Brave New World. All right, so let's talk now more specifically about genetics. So genetics, we know, as we said earlier, is the study of heredity, of how genetic information is passed on from parents to their offspring. So the person we sort of consider often the father of genetics is this gentleman. Gregor Mendel is his name. He was a monk, a religious person from Austria, Eastern Europe, not Australia, Austria. And so he was a religious person, but he had a hobby of doing experiments. And what he did is he raised pea plants. Okay? And he would crossbreed them together. So I've never grown peas at home. So peas grow on like a vine sort of thing. They produce flowers. Well, you can actually use a little paintbrush and take pollen from one flower and put it on the stigma of another flower and crossbreed them and see what the result would be. So he did that. And he did it thousands and thousands of times, carefully recording the characteristics that he would get when he crossed different types of plants together, when he mated them together. You would see what are the offspring like. What are those pea plants that are produces, what characteristics do they have? And he saw some things. Very, very careful logs of his data. Thousands and thousands of these crosses. We started to see some patterns emerging. So he had some conclusions. We'll just sort of simplify them a little bit. But what he concluded is that the traits he was seeing in these pea plants, things like the height of the plant, the color of the flower, the color of the pea, the texture of the pea, color of the pod. Each of those characteristics was controlled by two, what he called factors. Okay? One of them was inherited from each parent. And we know, we talked about this a little bit yesterday, that when the sex cells are created, each sex cell or gamete contributes one of its chromosomes to the offspring of each parent. So that's where these factors come from, carried in the chromosomes. So Mendel did all this work before anyone knew anything about DNA or genes or chromosomes for that matter. So what Mendel would do, he'd take these pea plants, get a little paintbrush. When the anther was producing pollen, he'd dust it off, get some pollen on the paintbrush, then he'd bring it to another plant, brush some of that pollen onto the stigma. Pollen would fertilize the ovules, he'd grow those seeds, and see what happened.
But did they ever, I mean, because you had to realize that the parents contributed.
Yeah, he realized they each parent
but did
contributed.
people ever realize? 'Cause
Yeah, they realized that parents influence the offspring, but never in this way. Never, because he came up with mathematical ratios about how this would happen. What's that? Yeah, that there are similarities, and he came up with more concrete mathematical ways of telling.
So like, say he wanted like a really tall one with like a lot of flowers. Would he take like the pollen from like a really tall one and like brush it onto the stigma of like a really
We'll get there, because he wasn't so much trying to produce a certain pea plant that was tall or short or whatever, but he was trying to say, okay, what if I take this tall pea plant and I breed it with a short pea plant? What happens with the offspring? We'll talk about that in 1 second. So we know, when we talked about karyotypes earlier, for example, in humans, each normal body cell contains 46 chromosomes, however, sperm and egg cells contain 23 each. When fertilization happens, and they form a zygote, how many chromosomes does that zygote have? 46. So what Mendel called factors, today we call them genes. Each parent contributes one gene to the offspring for the height of the pea plant, for example. And due to Mendel's work, we know that some versions of these genes are what we call dominant, and some versions are recessive. It's the opposite. So when we think about the dominant form of the gene, I also call them alleles, you can think of it as kind of like the stronger form. When we are in the recessive, is sort of the weaker form. It's the form that kind of can be hidden, can be covered up by the dominant if they're both present. We often use letters when we're talking about genetics, and we use a capital letter when we're talking about a dominant gene, a dominant allele, we call it. And we use lowercase when we're talking about a recessive allele. So for example, if we're talking about the gene in pea plants that makes them tall, we would use a capital T to indicate that gene. The tall allele is dominant in pea plants. If we have a recessive gene, we usually write it as a lowercase of the same letter as the dominant. We don't use a lowercase s for short. We usually use a lowercase t to show it's the other version. So if a pea plant has 2 genes for their height, one came from each parent, there's 3 possible combinations. What are they? What are the possible 2-letter combinations? Talia?
T.
They're all T's. Taya?
All three.
Yeah, well, give me one.
2 capital.
2 capital T's. Christopher? 2 lowercase t's. Or one of each. Those are the 3 possibilities when we talk about two genes. So we have some words we use for those. Homozygous dominant means you have 2 of the dominant genes. Big T, big T. Homozygous recessive means we get 2 lowercase. In the big T, little t, it's called heterozygous. That's a big T and a little t. We could write little t, big T, but the convention is we just always write the capital T first, and the order doesn't matter.
So when you're doing this, dominant is like a chain of the tall.
Yes, dominant covers up the other version. So let's think about it. This pea plant, if it had 2 big T genes, what would it look like? David?
It would be like really, like, that kind of like stretching out real long.
It would be tall. In this plant, obviously, short. Well, how about this plant? No. Why tall? Yes. So, this one is not medium in height. It actually is just as tall as this one. Because if there's one of these dominant alleles present, that's the one that kind of takes over. And so this recessive allele kind of gets covered up. So this would be tall, short, tall.
Does this also decide your gender?
No, because that's controlled by sex chromosomes. Remember XY?
Oh.
So that's determined by that. And there's not dominant recessive, it's just which ones do you have.
Oh.
So we use some terms when we're talking about genetics. When we're talking about an organism, we can talk about something called its phenotype. This is sort of how its genes are expressed. What does it look like? Its physical appearance, you could think of it as. For example, tall pea plants. When we're talking that it's tall, we're talking about its phenotype. You say you have blonde hair, that's your phenotype. If you have green eyes, that's your phenotype. It's how the genes are expressed. When we talk about an organism's genotype, we're talking about which individual letters, which alleles do they have. So when we said big T, little t, or heterozygous, or homozygous recessive, we're referring to an organism's genotype. So if we're thinking about the height of pea plants, what's the phenotype of this pea plant?
T. Tall.
Tall. What are its possible genotype?
T.
They're all T's.
Oh, capital T, capital T.
Capital T, capital T,
Or capital T, T.
or one of each. Both of those result in a tall plant. What's the phenotype of this plant?
Short.
Short. And its genotype?
Lowercase.
Yes, 2 lowercase.
Oh, I was,
Yes,
so is there more of a chance it's tall?
sort of. Not always, but we'll look at it. So this brings us, now that we know this, that for these characteristics in pea plants, they were each controlled by 2 genes, and there are 2 different versions of each trait. Because we know how these genes are inherited, we can start to predict what happens when 2 parents produce offspring. And we do it using a tool called a Punnett square. Punnett square is just a box with 4 boxes inside of it that we use to predict the results of a cross between 2 parents. So here's an example. If I had one parent that is big T, homozygous dominant, and I cross it with a parent that's homozygous recessive, 2 little t's, what are the offspring going to be like? So yeah, we can do the Punnett square. What we do, we take one of the parents and put their letters on top, one over each box. So we'll take this parent, we take the other parent's genotype and put it on the side. Little t in this case. And then I combine to fill in each box. So this box gets a big T from here, little t from there.
And you go top to bottom.
Big, doesn't matter, big T, little t. We always write the big T, the capital first for some reason. Here, big T, little t. Okay. And here, big T, little t. So, Mendel did this many times with several characteristics. He would take pure tall plants and cross them with pure short plants. And every single time he did it, every single offspring was what?
Tall.
Tall. He never got short offspring from that cross. Okay? So what we often will do is write the percentages, the probabilities. So in this case, we only end up with one genotype. These boxes each represent offspring, 25% of the offspring, because there's 4 boxes. So in this case, for this cross, 100% of the offspring are going to be heterozygous, big T, little t. And 100% are going to be what? Tall. Okay, let's look at this example. Here we're crossing a tall and a short pea plant. A heterozygous tall pea plant with a homozygous recessive pea plant. Same procedure, one parent goes on top, big T, little t. The other parent goes on the side, little t, little t. All right. Emily, what's going to go in this box? Emily, yeah, big T from the top, little t from the side. Emily Piccola, how about this? All right, let's go to this box. Big T, little t. Sandy, what goes here? Yep. And Mateo? Yes. All right, so now we have a different result. What would you expect in this, from these offspring? Talia?
Like 50% of it would be tall.
Yep.
50%.
How about in their genotype? Let's start with genotype. Because you were telling me tall, when you're saying tall, you're talking about their phenotype.
So probably capital T,
Okay, so there's 2 possible genotypes. Big T, little t, and 2 little t's. Heterozygous and homozygous recessive. What's the probability of getting either one?
50%.
50-50. So these are probabilities. They're not guaranteed. If I did this and I produced 100 seeds from these 2 parents, I would expect even numbers of each. Tall, big T, little t, and little t, little t. Now, is it always going to come out that way?
No.
No. Okay, sometimes you might end up with a little bit of a skewed ratio. But overall, the likelihood is that 50% are going to be tall, 50% are going to be short. David?
How could you tell in humans what their
Genotype is?
genotype is?
Sometimes. Depends on the trait. I'll talk about that in a minute. So let's do one last cross. Why don't you guys do this cross between 2 heterozygous parents? You fill it in. So one parent goes on top, big T, little t. The other parent goes on the side, So what is this parent? Tall or short?
Tall.
And this one?
Tall.
Tall. All right, so you take two pea plants, cross them, you're gonna get all tall plants, right?
No.
Wrong. This is something Mendel saw. Because what he did is in his first generation, he would do these pure crosses. Pure tall and pure short. And you get all dominant offspring. But then he would take two of these offspring and cross them. And he saw, well, even though these are all tall, somewhere in there is this short factor. Because when we take two of these and cross them, we get what goes here, Richard? TT capital. Dan, what goes here?
Big T, little T.
Yes, heterozygous. What goes here, Aisha?
Big T, little T.
Yes, heterozygous. Goes here, David.
TT.
Yes, they're all T's.
Little T.
Little T? One little T?
Two little T.
Thank you. All right, so now we end up with a little bit of a different ratio. So what's our genotype? What genotypes do we have here, Dylan?
50%, big T, little t, 25%, big T, wait, no. Yeah, 25%, two big T's and 25%, two little T's.
Correct. So we end up with this ratio, 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive. What's our ratio of phenotypes, Reagan?
Tall is 75% and short is 25%.
Correct. And so Mendel saw this. Every time he took two of these offspring and crossed them, he would end up with the same ratios. About 3/4 of the plants would have the dominant trait, and 1/4 would have the recessive trait. And he did the same process, but instead of tall and short plants, he did purple flowered plants versus white flowered plants. And he took pure purple, pure white, crossed them, and saw that the ratio, they were all purple. Then he took two of those, crossed them, and he again got the 3-to-1 ratio. And he did it with the color of the peas. And he did it with the texture of the peas.
So was the.
And all these other factors, and he continually found the same ratios were coming up. Right? And that's what led him to his ideas about, okay, well there must be two factors combining, one of them can be dominant over the other, and can be hidden, one can be hidden, but it can show up in that next generation if it's there.
Like, so which one would be the dominant, is that?
What's that?
Which one would be the
Yeah.
dominant?
Richard?
If the big T, little t was the same height as the big T, how did he know which one was?
He didn't. What he would do is he would to make these pure plants, he would repeatedly breed them until he never got any short ones, and then he was sure that because if you take this pure plant and you breed it with a hybrid, and you never get any short plants, then you're sure after a while that it's homozygous dominant. All right, let's look at just another example. Now we're on to a different trait. Now we're on the color of the peas. Peas can actually be yellow or green.
Really?
Yeah. You ever see like those yellow split peas, like they're dried?
I see.
You know, they sell them as sort of dried beans or yellow. So, how many people like split pea soup? Anyone? I love split pea soup. Nobody in my whole family likes split pea soup. It's really good. Anyway. And yesterday, or 2 days ago, I brought Miss Carpenter and Mr. Arcuri risotto that I made with peas in it, and both of them picked all the peas out and only ate the risotto. Like children. Anyway, so let's see here, we have a Punnett square. Do we have a yellow plant, a yellow pea that's heterozygous, and then we have the homozygous recessive green pea. We do our Punnett square. For this characteristic, we have big Y, little y, so we're not using T's anymore, because we're not on tall and short, we're on yellow or green. Put the other parent on the side. What do I get here? I'm calling you all outside. Dylan, what do we get here?
You get big Y, little y.
Correct. David, here?
We got two little Y's.
There. Big Y, little y, little y, little y. So what percent of the peas are going to be yellow in this example? Christopher?
50%.
Yeah, 50% yellow, 50% green. All right. Yeah, we're gonna watch it.
What do you have
It's just about peas. About
That one green one.
These days, scientists know how to compare characteristics for plants. They were able to calculate probabilities of having a specific trait getting a genetic disease according to information they have from the parents and family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century, to a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of genes.